This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1249, recorded on August 29, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from College Station, Texas, Jolene Ramsey.
Hello. Nice to be here. It's 91 degrees Fahrenheit here, and it's actually cooler this afternoon than it was last night at 9 p.m. I think we're going to get rain this weekend.
Here it is 25 C and cloudy in Chelsea. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Jolene, I got you beat. At least according to the Weather Channel, it's 102. I haven't looked at our digital thermometer. I need to text my wife and have her, who's out in the other room, and have her check that out and maybe report in. But clear blue sky. We're looking at, it's summer. But we're also looking at, I think this is the peak, and it may start to cool off. We're looking at, look at this, Sunday, 89. And a possibility of rain, same Monday. So that's all good.
See, we have like a 60 to 70% chance of rain and thunderstorms on Sunday and Monday.
Yeah. Okay. So obviously, you and I are proximal.
Mm-hmm.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is 75 and cloudy here. There's a lot of air conditioning inside, so I'm feeling very jealous of the warm temperatures that Jolene and Rich are telling us about.
I remember when we drove to College Station, Jolene, I got out of the car and this big baking air hit us.
Yes.
It's incredible.
Yes.
I don't mind it at all because I like the heat, but it was much hotter. What did we do? Where did we drive from? Your place, Rich?
Yeah. I think
right?
Was that coordinated with TWiV 500?
Yeah, it was.
We did TWiV 500 and then we drove out to College Station. Yeah.
Other way, I think.
Other way?
Yeah,
we did theirs first. We did 501.
It's actually kind of a nice drive. It goes through some pine forests and stuff like that.
You went through Bastrop.
How far was it? I don't even remember. 2 hours? Do you remember?
Yeah, it's about 2
about 2 hours.
And then we stayed over and went back the next day, right? Something like that.
Mm-hmm. That's right.
What year was that, Julie? Do you remember?
2018.
Yeah, it was. TWiV 500 was on my 70th birthday.
That's cool. I think you came to 500, right, Jolene?
I did.
Or
yep.
No? You did.
Me and my friend Justin Levitt, we were fellow postdocs. We may or may not have been pulled over on the way.
Well, if you like these podcasts about science, real science, no lies, no misinformation, no nonsense, or to paraphrase Stanley Plotkin, no people who don't know what they're talking about. Although sometimes if I talk about phages, I don't know what I'm talking about, but we have Jolene here, so we take care of that. Anyway, if you like these science discussions, we'd love your support to support them financially, because we don't do ads. We depend on your support. Whenever I post a YouTube video, there's a box that says, do you want to monetize or not? And I say, no, don't want these ads interrupting. I know a lot of people are used to ads, but don't want it. Can you imagine lecturing in the middle of a class and then I stop and give an ad for something? Well, actually, the students might like that, depending on the ad. Anyway, microbe.tv/contribute. We'd love to have your support. We have one news item today, which we cannot not mention because everyone probably knows about it. I picked a CNN article, but there are plenty of others. Of course, this is all about the CDC director, Dr. Susan Monterez, who RFK Jr. asked her to quit because she wouldn't agree to accept everything his ACIP concluded and to stop saying vaccines were good, so he fired her. And then because he can't fire her because she's confirmed by the Senate. So only the president can fire her. So he fired her on X, and she said, that doesn't count. You got to send me a letter. So we're at some kind of an impasse right now. I don't know what's going to happen, but it's really crazy.
I think a lot of other people at CDC also resigned sort of in protest of her treatment.
That's right.
Yeah.
Actually, RFK wanted her to fire some people at CDC, and she said, no, not doing it.
And I think I'm actually enthusiastic about this whole thing. I think that she and the ones who are supporting her by resigning or whatever are enormously courageous and doing the right thing. This is a confrontation that has to happen. And my guess is that much like the people who left or were fired from the ACIP, we will see satellite organizations that will rise up and make appropriate recommendations. What will happen to that? Who knows? But this is what's going on is completely outrageous. I want people to understand that. It's insane. This is insane. Okay? And the people who are standing up to it are doing exactly the right thing. The guy who's been suggested as to replace the director is the current deputy director, named James O'Neill, who is an American science and technology investor, who has degrees from credible schools, Yale University and University of Chicago, a bachelor's and a master's, respectively, in humanities. So he's a humanities-trained, liberal arts education-trained tech investor who has absolutely no credentials whatsoever to be in this position. None.
That's what you need to be in a position in this administration.
Absolutely.
No
you need fealty to the President of the United States. And that
it's a good word.
That brings with it an interesting collection of nonsense and delusion and cruelty that seems to come along with that.
Brianne, fealty is a great Jeopardy word.
It really is. It really is. Fealty is a wonderful, wonderful word. I also would like to, defend liberal arts backgrounds and humanities. We do lovely things. We do not necessarily support fealty. And the key thing here is that for a position in science, you should have some science training.
Yeah.
There are plenty of other useful things one can do outside of science with the training that he has. This is not the appropriate training for this position.
Fealty, by the way, is an old-fashioned word that means intense loyalty, fidelity, or faithfulness, like to a ruler or a lord in a feudal system. They owed fealty to the earl rather than the king. So apparently, her lawyers, Monterez's lawyers, said, only the president can fire her, and they got the notification from White House staff in the personnel office that she was fired, and they said, sorry, it doesn't count. It has to come from Trump. And so RFK also wanted her to fire other people, Deborah Houry, Dimitri Daskalakis, and Dan Jernigan, and she said no. And he's about to release next month The Cause of Autism, right? He's researched it for a month, and she didn't like that. She was saying, I'm not going to support anything that you say. So this is where we are. And it is crazy, as Rich said, this is totally not the way a government is supposed to work. And there's a reason why the U.S. government has evolved to be the way it is, because it works very well and has respect. But now the respect has ended.
I wonder if his cause for autism that he's going to find next month has any relationship with his messaging, I guess, to the journal.
Yes.
Who that published the study about vaccines and aluminum in Denmark that we covered in a previous episode. He has personally asked the journal to retract that paper, and they have said no.
Just because.
Yeah.
I think that's probably an indication that it's going to be something to do with the aluminum. Yeah. Oh man, it's going to be disgusting.
Well, I just take heart that people are in a very, very public, prominent fashion pushing back. And not just in this realm, in other realms as well. The attempts to hire the fire the Fed, I forget what the position
chair,
is. Not the chair, but one of the people on the board who said, no, you can't fire me. I'm not leaving. Good.
Also, so it turns out that, the medical societies are saying, no, these vaccine recommendations are wrong.
Yeah.
There are at least 2 and probably more that have said, we recommend this and this and this. COVID vaccine should be given to people under 65 and pregnant women, for example. So that's good. But it's going to be a little confusing to people, right? Because most people don't know what a medical society is.
Yeah, that's the problem. It's going to be confusing, and it's going to sow distrust in, right now, appropriate distrust in the government agencies. But the problem is that when all this is over, I hope, it's going to be a project to restore that trust.
Mm-hmm. I think it may also, at least in the short term, influence availability and things like insurance coverage.
Yeah. Yes. Insurance coverage is probably one of the biggest deals, By the way, my wife texted me that it's 100 degrees according to our outdoor thermometer.
I just wanted to mention, if you haven't heard Daniel Griffin's clinical update, which pre-releases this episode by 24 hours, we talked about the Legionella outbreak in Harlem here that's been ongoing. And the New York City Department of Health has been checking cooling towers for Legionella bacteria because that's typically how it's acquired. And they have, first they look by PCR. They get some water and they do a quick PCR, right? And then if it's positive, then they do a culture test. And so Daniel said, they check the tower, they do PCR. This DNA may be from bacteria that are alive or dead. Mm-hmm. And then he said, dead bacteria cannot make people sick. And so then they do culture positivity, and they found 12 cooling towers that have live, and we can say live for bacteria, live Legionella bacteria in them. And then they remediate them. They take all the water out and they decontaminate them and so forth. So it's very interesting. I like that.
I would
bacteria.
Object to that statement. Can't, Clostridium botulinum toxin, they are no longer alive when the toxin makes you sick, right? Isn't that how
I think
contamination occurs and the bacteria grow, but then they make the toxin and they die and at some point later?
Technically, it's the toxin, right, then?
But if you did the PCR, then you would detect the DNA.
Yeah, you would detect the dead bacteria. But, I mean, it's a little bit of a technicality, right? Because the bacteria have already made the toxin, right?
True.
I don't know. But anyway, it's a cute statement, nonetheless.
Yes.
It's probably mostly true. Maybe it's 99% true. It's like this what is the statement? Dead men
good
tell
enough
no
for
tale.
Government work, let's
say. Good enough for government.
Wow. That's being totally reevaluated right now.
Yeah.
Okay. It is time for some papers. Now, the snippet is an article in a journal that I probably have never looked at in my life, the Journal of Alzheimer's Disease. Maybe I should, because I'm 72. Who are you again? TWiV. I was, the other night, I couldn't remember Stanley Plotkin's name. And I said, I can remember all the viruses and many other things, but for some reason, people's names evade me. Anyway, this Journal of Alzheimer's Disease. Herpes simplex viral infection doubles the risk of dementia in a contemporary cohort of older adults, a prospective study. It's actually not a joking matter, but I came across this paper when we were doing another one recently, 2 weeks ago, which referenced this paper. And so I thought it would be interesting to look at the evidence that herpes simplex viruses are thought to play a role in dementia onset. Okay, so this comes out of Sweden, Uppsala University and Umeå University. Umeå, right. Erika Westin, Gustav Bostrom, Jan Olsen, Fredrik Elg, Lars Lind, Lena Killander, Hugo Lovheim, and Bodil Weidung. So 55 million people had dementia in 2023. Nearly 10 million people a year get dementia diagnosis. And the big one, the most common cause of dementia is Alzheimer's disease, right? There are all different other kinds of dementias, but Alzheimer's is the big one. And many studies, or multiple studies, I should say, have suggested a role for herpes simplex virus infection in either Alzheimer's or dementia in general. So as you may know, if you've listened to TWiV, there are two kinds of herpes simplex viruses, type 1 and 2, that infect humans. They're generally associated with orofacial and genital infection, although there can be some blurring of those lines. And if they're studied separately, HSV1 is the one associated with dementia risk.
And it's the one associated with above the waist oral lesions.
Yeah, orofacial lesions. So
cold sores, dudes. Cold sores.
Cold sores. In Sweden, 80% of adults are carriers, which means they're antibody positive. And you can look for IgG or IgM. So IgM typically arising on a reactivation. And so the Alzheimer's or dementia risk has been associated with both IgG and IgM prevalence in previous studies. Quite a few studies have been done. And in addition to this link between herpes simplex viruses and dementia, there's also a link between a cellular gene called apolipoprotein epsilon 4, ApoE epsilon 4. And this may be the strongest risk factor for Alzheimer's disease, and maybe in combination with herpes, it multiplies, and maybe something else as well. So many studies have found an association between herpes simplex viruses and dementias, but others have not, right? Others have looked at IgM and IgG to herpes simplex viruses. They've found no link. They have found no interaction with ApoE epsilon 4. And that's the problem, right? When you have this uncertainty in the literature, that maybe it's because these are observational studies, that you're just looking at populations of people and their confounding factors, or maybe your study population is too small and so forth. And that's something they try to address here. The other thing that has been found is that if you treat herpes virus infection with an antiviral, it may reduce the dementia risk. And in one study, it reduced risk by 70% in people who had antibody to HSV1. And it also reduces dementia risk in people with symptomatic infection. However, symptomatic infection. But other studies have found no link between treatment and dementia risk.
So to summarize, sometimes the answer is yes, they're linked, and sometimes the answer is no, they're not linked.
Right. So what do you do? Yeah. What do you do is the question. You keep looking, right?
Yeah.
Because you can't give up.
Yeah. So I was looking at this paper and I thought it was really interesting. And I also thought about some past discussions we've had about similar things. So in TWiV episode 519, there is a discussion about herpes simplex viruses and HSV1 and Alzheimer's disease, and specifically also looking at sort of ways that some of the things that happen in Alzheimer's brains may actually be antiviral, and so perhaps it makes sense that we see this. In 1207, there was discussion of a recent paper that came out in Nature looking at how vaccination with Zostavax, which is not for herpes simplex viruses, HSV1 or 2, but it's for a different herpes virus that is varicella zoster, the causative agent of chickenpox. That vaccine in a very large study seemed to protect individuals from dementia risk. And so both of those types of studies kind of went along with this. And I was also really reminded, and part of the reason why I point that out, and I think it's really interesting, is if you look at this paper, they looked at both HSV herpes simplex viruses, and they also looked at cytomegalovirus, yet another herpes virus, and they said they saw no link with cytomegalovirus. And so it seems like it might be some herpes viruses, but not all, and it sort of complicates the study further. In looking at all of this, I definitely am reminded of sort of the literature about Epstein-Barr virus, in particular with Epstein-Barr and multiple sclerosis, where there was this same exact kind of, there kind of is, but there kind of isn't, and I don't know, thing. And part of the reason that that's really hard is because so many people get Epstein-Barr early in life. And so maybe it's like if you got it early versus if you got it late, or when your reactivation happened or things like that. And you need a really, really big sample size to figure that out. And we talked on TWiV 869 about an Epstein-Barr MS study that used a crazy large sample size in order, and really like finally got at it. But they had to use like the U.S. military recruits data, and they had something like 10 million subjects. Because they were having such a hard time finding people who were actually getting that infection for the first time at certain times in life, and being able to sort of measure some of those things. And so I think this is fascinating. I think that there's a lot of evidence pointing in the direction of something in the herpes virus group being involved in dementia. And I really want them to be able to start to pick at how frequent was reactivation, or when did you get infected, or all of those types of things, because that might be confounding this and making it kind of muddy.
It's not just an antibody positivity, which you'll see here. It doesn't matter how much antibody you have. It's just whether you have it or not against the virus.
In my mind, it's relevant that the herpes viruses that have had some association with Alzheimer's are neurotropic.
That's right.
Okay.
That's right.
So these, just to remind people, these are viruses that at least HSV1, for example, establishes an infection in epithelial tissue, but then takes up latency in neurons and can be reactivated in neurons, ordinarily in the peripheral nervous system. But there are complications where herpes type 1 can infect the brain and cause an encephalitis. So it's not an unfamiliar hangout for them, as opposed to cytomegalovirus, which is not specifically neurotropic, right? Has tropism for other tissues. HSV2 is the same way. That's also neurotropic. VZV, the varicella zoster virus, is neurotropic.
What's interesting is that, herpes encephalitis is a relatively rare event, 1 in 250,000 herpes infections, but it must be more frequent than we know if it's contributing to Alzheimer's. There must be many incursions of the brain by herpes viruses.
Yeah, a glancing blow.
Yes.
Do you mean like it gets there and then somehow it's cleared so we don't see it there or don't know that it's there? Or
yeah, so as Brianne mentioned, plaque formation may be a response to infections. And so the virus may come and go, but the protein is made. What is the protein in the plaques, Brianne?
Amyloid beta.
Amyloid beta, and then it's made and then somehow the infection is cleared quickly before you get pathology. But then this amyloid beta is there, and if you get repeated infections, you get more and more deposition, and that could be a risk factor. But
And an
one
important thing, I think the multiple sclerosis thing is an important example of how you ultimately nail this kind of stuff down. And an important component of the multiple sclerosis thing is that whether or not there's, your genetic background, the patient's genetic background is an important component of this. All right. And so teasing that out is another whole problem itself, and it may play into this.
The key is that these two studies that Brienne mentions, the Epstein-Barr in the Army and the Zostavax and dementia, took unique approaches to contribute where everything before that had been confusing.
Mm-hmm.
The Epstein-Barr in the Army, they get people at a young age and they watch them seroconvert, and then they can follow them and see who develops dementia, who does not. And then the other study, they had people who just turned 70, so presumably they have similar risk factors, and those are the ones admitted to the study. So it's the same. It's going to be the same with herpes simplex viruses and dementia. Someone's going to have a unique approach to figure it out, because even with CMV, I mean, the CMV is less convincing because 1 of 9 studies found an association with dementia. So, and because it's not known to be a neurotropic virus, it's probably not. But the herpes, it looks like it's 50-50. And so something's going on there. And so that's why people may think, well, how in science do you know if you've done 20 studies and 10 say one thing and 10 say another? How do you know? Should you keep at it? And how do you do it? The key is there's something there, and you have to find a unique approach to answer the question.
Yeah. And so, as I was reading this, I was kind of thinking to myself, I'm really interested in sort of the future paper that is similar to that Zostavax dementia paper or the Army EBV paper, because it seems like that's what's really going to be needed here.
I mean, they point out in the paper, they say, so we do not know the relationship between HSV and dementia. The results are far from unanimous. Okay?
Yeah, they're really good about doing the background on this and saying it's confusing.
And at the beginning and at the end.
That's right. Mm-hmm. And then the middle is confusing because it's a lot of statistics, but we'll make our way through it. They also say that age is it's the strongest risk factor for dementia, right? How do you adjust for age? Everybody's getting old. And so how do you adjust for that as opposed to a herpes simplex virus infection? So, they try and do that by getting people who are the same age, but the other studies did not. So here they're going to look at the role of herpes simplex viruses, which would be both, and herpes simplex 1. They're going to stratify the data, and then CMV in Alzheimer's and dementia risk. And they're also going to look at the role of ApoE epsilon 4 carriership and herpes virus treatment in a cohort of same age individuals. So that's the key. So this is a cohort in Uppsala, the prospective investigation of vasculature in Uppsala seniors. All right. It was not designed to address dementia. It was designed to look at if it's worthwhile if it's easy to use a measure of endothelial function and arterial compliance to prevent future vascular events. And so basically they invited 2,000 people in Uppsala within 2 months of their 70th birthday to participate in that study. 1,000 people did.
And that was initiated in 2001.
2001.
Right? So
that's
25
right.
years ago.
And then,
so it's a prospective study, meaning they had a goal, they recruited people, and they followed them over time. And amazingly, they had over 50%, or is it just under 50%?
Yeah, that's
50%.
Participation. It's pretty good.
And then they would be assessed at 70 initially, then 75 and 80. And so directly, and then at 85, it was through their records, so they didn't have to come in anymore and get assessed. So that's the study population. They took serum so they could measure antibodies to herpes simplex viruses. They had, and some of these individuals, of course, developed dementia over the years, and they had a specialist collect the information on dementia and Alzheimer's diagnoses, right? And then they also had information on whether they were treated for the herpes virus infections. And the treatment is defined as a prescription of at least one anti-herpes drug before the outcome, or what they call censoring, which is, I think, the person dies.
It's they die or they are lost to the study, so they decide not to participate anymore.
I think it's an interesting use of censoring.
It's one of those statistician things.
Yes. Okay, so what did they find? So they divide all the individuals into subsamples. So they have, in terms of antibodies, they have anti-HSV, which would be 1 and 2, and then HSV1 only, and then anti-CMV, defined by IgG seropositivity. And then they want to look at the effect of treatment and the onset of dementia, of course. They normalize for educational level. So you may ask, why is that important? Well, the idea is that if you have a certain education, you'll know that things aren't right if you're beginning to lose mental capacity, and then you will seek health care, right? Whereas if you don't know that dementia is a thing, which is possible, you may have symptoms and just keep on living and deteriorate, and it's not useful for this kind of study, obviously. And then they do statistical procedures, of course, to look at risk factors and so forth. So 1,002 participants in the studies. 71 of them developed dementia of various causes. That's 7%. And 36 of them, 4%, developed Alzheimer's disease. So I think this is a relatively representative population. So that probably is a good indication of AD risk or dementia risk worldwide. So 71, 100 out of 1,000, about 10% risk of developing dementia after 70. Is that a reasonable?
I don't know that much about who lives in Uppsala, Sweden.
Yes, that's right.
Well, so maybe representative of the whatever kinds of populations live there.
Yeah, I'm sure that others have done more accurate estimations than that, but I'm just thinking 10% seems about right. Anyway, the prevalence of IgG, anti-HSV IgG, was somewhat greater among persons with Alzheimer's disease and dementia than among those without dementia. So we got 89 and 89% positivity with dementia and 82% without. 6% of the participants got anti-herpes treatment up to the last 5 years before the ends of follow-up. And this sentence is important. The proportion of participants with Alzheimer's disease and dementia who had received anti-herpes treatment at least 5 years before diagnosis were larger than that of participants without dementia who received anti-herpes virus treatment at least 5 years. Right? It's a little bit. You may have to.
It's hard. I had to
think about
read it like 5 times. It's highlighted.
Yes. Proportions of participants with dementia who had received anti-herpes treatment were larger than that of participants without dementia.
So that's linking virus with dementia again.
Yes.
Because you're more likely to have been treated if you had the virus.
That's right. Okay, so then they have a figure which shows the time to dementia diagnosis according to whether you have IgG against herpes, HSV, or not. So basically, and here's the one thing you can listen to if you don't want to listen to anything else. Anti-HSV IgG positivity doubled the risk of dementia in this population, in this study. And they give hazard ratios as models for that, as numbers for that. And that's in the HSV subset of patients. But when they put everybody together, you don't see that difference. Interesting. They don't find any interaction between anti-HSV IgG and ApoE epsilon 4 positivity. So they don't find any interaction between that particular gene that's been found before. And so it's whether or not you have IgG that doubles your risk of dementia. But if you have higher titers, that doesn't matter. You don't have any greater chance of having dementia if you have higher titers of antibodies. There's no connection at all. And same with IgM. So anti-herpes virus drug treatment was not associated with a lower risk of Alzheimer's disease or dementia. Antibodies to CMV were also not associated with Alzheimer's or dementia. And then they added other diseases that people of this age are likely to have: stroke, myocardial infarction, diabetes. They get the same results. So those are not confounding the results. That's it. That's the story. I think if you are seropositive for HSV, you double your risk of acquiring dementia or Alzheimer's disease.
So this confused me, and what I am going to say, or quote, confused me, and I want help. So they say anti-HSV IgG positivity doubled the risk of dementia in the basic and fully adjusted models. Next sentence. No significant association between anti-HSV IgG positivity and Alzheimer's disease was found in the full sample. Right. So I take from that there's a distinction in this study between dementia and Alzheimer's disease. They didn't find if I read this correctly, they didn't find a specific association with Alzheimer's disease, but they did with dementia. And I take it that these are two different subsets. I mean, yes. Alzheimer's is a form of dementia, but if you had Alzheimer's, you're not in the dementia pool they're talking about, right?
Yes.
So am I looking at that correctly?
Yeah, that's right. And they say it's because the sample size was small and probably couldn't pick up the Alzheimer's signal, right? Yeah.
Right. And I think that some of the ways that some of the criteria for Alzheimer's disease are sort of challenging to measure. Mm-hmm. And so sometimes there'll be patients who have some memory impairment, but may not check those boxes, or you may not be able to do the studies needed to figure out if they check the boxes. Mm-hmm. And so that, I think, is also a really sort of something that is going to have to go into those larger studies, is a really careful definition of Alzheimer's disease and dementia. Because I think that different studies might be defining those in slightly different ways.
Yeah, so my understanding has been that the only definitive diagnosis of Alzheimer's is post-mortem, right?
Correct. That was my understanding too.
Yeah, okay. Yeah.
So by the way, the global risk of Alzheimer's is 10% at age 65 and up. So looks like Uppsala is somewhat representative in that sense of the rest of the world. But yes, I mean, you can't always extrapolate. So the discussion is very good. They have a lot of good points here.
I think, one of the things that they mention is something that we were discussing at the beginning, is that the non-significant interaction between the previously linked ApoE epsilon 4 and anti-HSV IgG was probably related to their lack of power. Right, we're talking about millions versus they had 70-ish people here that developed dementia, and they started with 1,000, which sounds like a lot, but in the end, it's actually not enough to really pull out some of these details that they're interested in knowing.
Yeah, it's going to take the studies like what Brianne was talking about, in a study population in the millions to really nail it down.
Well, there are millions of people around the world just having bodies and cooperation who can coordinate something like that, and sufficient data to know the dementia versus Alzheimer's disease or other things that are going to influence this final outcome.
Yeah.
We just don't have that right now.
Yeah. And so I guess all of those things make me not totally surprised that it's a little muddy.
Mm-hmm.
Yes, sure.
It's going to come out of databases from national health programs or something like that,
Yes.
or military for, veterans affairs or something like that.
So they point out that they have controlled for age in this study, right? Because they started everyone at 70 and they're all progressing at the same rate, as far as I know. That's how age works. And so the age confounding factor is removed from these studies. And they also point out that, I think I said this before, their study was too small to identify an effect with Alzheimer's. And they say most of the dementia in this study were probably Alzheimer's, but they weren't always diagnosed as Alzheimer's, right? That doesn't always happen. And also they say the lack of interaction between HSV, IgG, and ApoE epsilon 4 is probably due to lack of power. Not enough subjects in this. There was one thing that they talked about and they didn't explain. So it has to do with IgM, I believe. What was that? Lack of association between IgM seroprevalence and dementia risk. Not sure why that would be. Right? Brianne, any ideas?
I mean, I think that that might have something to do with the population's age and versus when they first encountered HSV.
Mm-hmm.
It's probably pretty likely. I don't know the data offhand, but I think most people are coming in contact with these herpes viruses relatively early in life. And so the IgG likely came from things like reactivations and not an initial infection with HSV. And so most of these people are probably, too far away from their initial infection for IgM to be particularly relevant at this point.
So IgM would only be seen in that first infection. A reactivation would not give you IgM.
So you're going to have more of the memory cells.
Yes.
And you're going to be making IgM. Yeah. Or sorry, IgG.
I remember reading, though, they said that IgM is also a measure of reactivation, but I don't know. I don't understand why they were saying that.
I don't know these data well enough to know.
IgM. Let's see. Let me see if I can find it here.
While Vincent is looking, I want to point out to the listenership that this is science in action here. You hear a lot of people saying, I don't know. I don't understand. It's not clear. This is how it's supposed to work.
HSV infection can be demonstrated, they call it carriership, can be demonstrated by the presence of IgG in serum, and IgM and IgM detection and higher IgG levels reflect frequent reactivation. So I don't understand why, because it should be mostly memory on reactivation, right? So maybe they don't know. I don't know.
Well, maybe you reactivate frequently because your memory sucks.
Maybe.
Yeah, it could be. Anyway, there's clearly an association between herpes simplex viruses and dementia, but what else is going on, we don't know.
Stay tuned.
Go ahead.
So HSV1, is it very different are there like strains of HSV1 that are more prevalent in different areas of the world? I don't know that much about it.
There's certainly strains, but I don't know about geographic prevalence.
I can't answer that.
Don't know that at all.
I don't think of it as being super variable.
Right.
Yeah, it's not like one vaccine should work against one, right?
Mm-hmm.
And one should work against two. I don't think you're going to have cross-reactivity, but I could be wrong. One thing that I forgot to mention, they say in the discussion, what we need is a placebo-controlled trial of a herpes antiviral, right? So you take people, and this is one of the places this is happening is at Columbia, because I remember reading about it a couple of years ago, and I knew someone who tried to get into it. So they take people at this age, 65-ish, where you're going to have dementia onset within the next years, and they do MRIs to see if they have early onset Alzheimer's, right? And then they take those and enroll them into a trial where they were giving them valacyclovir, oral herpes virus drug, and then they would follow them for many years. And so that study is ongoing still. We don't know the results of that. And the idea would be, if you have early onset, you don't have terrible symptoms yet, and eventually they get worse and worse. And if the drug works, it's going to ameliorate those symptoms. Well, that would be the idea anyway.
I wonder how early you'd have to get the drug in order to really prevent the reactivations.
Right?
Yeah,
that's the question. I don't know how else you could do it. You don't want to put someone on valacyclovir from 60 on up, right? That's no good. You don't want to do that. Although it seems to be a relatively safe drug, because without herpes virus infection, it doesn't do anything, right? It needs to be phosphorylated by a herpes virus thymidine kinase. So
I know that it's over the counter in other countries.
Oh yeah,
not here, yeah. You can get it over the counter in Europe, which I sadly know because one of my friends tells me that every time I go to Europe, and asks me to bring some back.
So it would be very interesting if valacyclovir. So valacyclovir is a modification of acyclovir, which makes it orally bioavailable. The same mechanism of action. It'd be very interesting if that worked, because then what would be the therapeutic modality? What are you going to do? Get people at 65 and have them take it like vitamins, right? I don't know.
People would, if it was going to
sure
reduce their risk or almost eliminate their risk of developing dementia. Would you? Wouldn't you?
Yes, but it's not going to be 100%. I would certainly take it, but there are other things that are going to cause dementia as well, right? I mean, it may be half. I don't know what the fraction is going to be. Same with Epstein-Barr and MS. It's not all MS caused by EBV, but you take your chances. Yeah, I would do it. I would take it because it's a really safe drug. And that's why it was developed, I think, in the 70s. And it's the only drug because nothing can compete against it. It's so good. Other candidates have been developed, like they made an inhibitor of the helicase primase of herpes viruses. The company said, forget it. We can't compete against valacyclovir. Anyway, yeah, I would take it. It's a good drug.
By the way, speaking of, I can't resist this. Speaking of over the counter, the Texas state legislature recently passaged House Bill 25, making ivermectin available over the counter. In a statement from the Lieutenant Governor, Dan Patrick, it says, quote, "The COVID-19 pandemic has emphasized the need for increased medical freedom in Texas." Blah, blah, blah.
What is that all about? If you got vaccinated, you were okay. What's the problem? So
I just thought I'd stir you up a little bit, Vincent.
So ivermectin has been through multiple trials. It doesn't do anything for COVID. Why can't the legislature base their conclusions on science?
Is the legislature worried about parasite infections in Texas?
Who knows? But
we're worried about the new world screwworm.
Screwworm,
does it work against that?
Don't know. Probably not.
That's a good question. It's a good question. Look, it does.
Despite the name, it's actually a fly, but it is I think it's the larval stage that's the problem.
Yeah, they lay their larvae in a cut or wound, and then they burrow in and eat. Yes, ivermectin is effective. It's an insecticide. Yes, so it's effective. Many insects are susceptible to ivermectin. Right?
Ranchers take note.
Channels that are inhibited. Interesting. But the idea that a legislature would do this in the absence of any data that says it's beneficial, it's all that's going to do is cause harm, right? Actually, you could get it you could go into a farm supply store and buy ivermectin. It's a horse paste, right?
Mm-hmm.
Yeah.
And people did that during COVID.
People I know. I
Were
am
they
in
okay? Did they
Texas.
get sick? Some people got sick from having veterinary ivermectin. All right, let's move on to, if Jolene is here, we're going to do a phage paper, right? If it's like, if this is Tuesday, it's Belgium. If Jolene is here, we're doing phage.
Yep, we are doing another phage paper. So I chose a paper that came out this year. Well, it's a story I knew about before. This is from Cell Host Microbe. It's in Cell Host Microbe, and the title is "The Prototypic crAssphage is a Linear Phage-plasmid." The authors are Danica Schmidtke, Angela Hickey, Jacob Werble, Jordan Lin, Ivan Liachko, Gavin Sherlock, and Amy Bott. And these people are primarily working at Stanford University in a few different departments. And there's a representative from Phage Genomics in Seattle, Washington. So the big idea in this paper is that there are some phage that are incredibly abundant in the human gut, and we don't really understand how they are maintained there. And we can't culture all of them. So we're trying to understand just some very basic biology of their infectious cycle. How do they exist in the gut? And when I say how, I mean, what does their DNA look like? Is it in a capsid? Not so much. It's in the cell. So how is it in the cell? How is it maintained in the cell? And then the idea then being we can understand the ecology between the phages that are in the human microbiome and the bacteria that are important for maintaining a healthy gut. And here they were studying one, and they found that it exists as a plasmid, which is primarily a circular DNA molecule separate from the chromosome of the host bacteria, and it doesn't make a lot of phage in terms of what we're usually talking about when we're talking about viruses. It doesn't make a lot of particles that move around from cell to cell. And since this was one that is not previously culturable, they had to do this with a lot of indirect methods, but they eventually were able to determine some of the bacteria that it replicates in. So that's the kind of overview. Now, more in detail, this is about a phage that was originally called the prototypic crAssphage. And crAss originally came from the words cross assembly. So back in 2014 and some prior, people were looking at all this huge number of metagenomic reads, DNA that have been sequenced from the human gut, and they assembled from a mixture of those samples a genome that looked like it was a phage. And since it came from pieces that were put together from different samples, that was the cross assembly part. And this one is called the prototypic crAssphage because it was the first one, and it was very interestingly found to be in about 70% of all of the metagenomic samples that had been taken worldwide, which is a lot. And within individual metagenomes, so a person or a stool sample from a person, that it would reach greater than 40% of all the reads in an individual sample. So it was all over the world and very highly abundant within individuals.
I'm sorry, go ahead, please.
Jolene,
Yep.
So with the crassphages, already what you said is like super helpful. But if we think of that, of like crassphages as a group or others,
Yep.
Are there other types of similarities? Is this just DNA sampling, for example, or are these just phages we know because of metagenomics, and there's not necessarily other things that they have in common?
So the prototypic crAssphage was the first one. And after that one was assembled, then we looked for similar genomes and found that there were others that were related. They have a similar size, similar genome arrangement, some shared proteins, but they are quite variable. So this has now become an order of Crassphoralis, and their prevalence is varied across the globe. Most of them are not cultured. They are only detected through sequencing, but a few have now been cultured. When I say a few, it's actually like more than 30, but if you group them based on how similar they are to each other, it's really just a few. And part of the reason they haven't been cultured is because we don't know what their hosts are. So you can't make a plaque. And also, for the ones where we have been able to culture them, when I say we, I mean other researchers, they actually don't make plaques. They will grow on a plate, but they don't make that nice clearing that we love that makes it really easy to track them over time. And part of the tricky thing here is that most of these are gut anaerobes, so they're not as easy to work with in the lab as an E. coli. And then we just don't grow a lot of them. They grow a little bit slower. So that's, does that answer your question, Brianne?
It does. The only other piece I was wondering about is that, are these normally DNA phages?
Yeah.
Okay.
All the crassphages are double-stranded DNA phages of about 100 kb.
And most phages are DNA phages. Is that a correct statement?
Most known phages and cultured phages are DNA phages, but RNA phages are a lot more abundant than we used to think.
Okay.
We just weren't looking for them in the right way. And so there are a lot more of them out there. It's just that most of the studies and most of the way that we isolate them is biased towards double-stranded DNA phages.
So if I do metagenomic sequencing and I get out a contig, something that looks like, a piece of DNA that is unto itself, how do I decide that that's a phage and not a plasmid? Not a non-phage plasmid. What distinguishes it as a phage?
So the structural genes are a big part of it, as well as the lysis genes. So a phage is going to be a virus that makes a protein capsid that will carry its genomic material from one cell to another. And it has to have a way to get out of the cell. And since most bacteriophages will lyse open the cell at the end of their infection cycle, they'll have some sort of lysis genes. That's cool. That would be the difference, I would say. But here we have a mixture of a DNA molecule that has those genes, but it also has the genes that are quite characteristic of plasmids, which are things like moving the DNA from cell to cell without using a capsid through conjugation.
Jolene, what came first, a plasmid or a virus?
No one has ever asked me that question before. So
I'm like a pesky undergraduate.
I'll ask you what I ask my undergrads, which is, I don't know, what do you think?
Well, I think logically, plasmids probably came first, and then they stole a capsid from the host and became a virus. That's what I would think.
And what was the host using the capsid for?
Well, it was a structural protein of some kind that was repurposed, because that's what we see for eukaryotic viruses. We can see distant homologies in the structural proteins with cellular genes.
Mm-hmm.
And so that's the hypothesis there. So I think this idea that plasmids were probably there first. It's very simple to get into a cell and reproduce.
I could agree with that.
Steal a gene. So do the students ever have good answers of what they think?
Sometimes. Yeah. Some of my graduate students have better answer than my undergrads, but there are some very intelligent and deep thinking undergrads out there too.
Well, this is the thing. When you're not intimately associated with a field for a long time, you tend to have disarming questions, right?
You can think outside the box and outside the assumptions.
Yeah.
I was thinking about it too. I was like, so which eukaryotic viruses do I know that exist as episomes? And that was the herpes viruses do that. They're attached to the chromosome, but not part of the chromosome. I don't know if there are any other ones.
Yeah. The only other thing that I think of, and I honestly understand this so little that I don't even know all that much, is aren't some of the mycoviruses that infect fungi plasmids?
Yes. I believe that's the case.
That's
Good
right.
point.
They never leave they never leave the. Some of them don't even have a capsid. They never leave. But some of them actually have capsids, but they don't leave the cell either, which is weird. Yeah. I think there are other DNA viruses, Jolene, that exist as episomes.
Oh,
like
yes.
Hepatitis B virus, closed circular DNA,
sure.
besides the herpes viruses, and a bunch of others also. Yeah. So I think this plasmid virus thing is old and certainly has had influences in the eukaryotic world.
Well, and if it's linear, then parvos too.
Right.
Oh, okay. Yeah, that's right. So within this study of phage and plasmids, they talk about how recently we've kind of made this association, or there have been some measurements that have looked at what is a plasmid, what is a phage, and what is a phage-plasmid, as they call them. And one of the things that they talk about is that phage-plasmids are large DNA elements. They say that they have a 67.8 kilobase median size, whereas the median phage is 48.5 kilobases and the median plasmid is 59.1 kilobases. And another thing that they mention is that the plasmids tend to have independent origins of replication, which, I mean, a phage also has an origin of replication, but there are certain types of origins of replication.
So, Jolene, when I was young and I learned bacterial genetics, there was a word, phasmid, which was a phage plasmid. Do they still use that?
I'm more familiar with the term phage-mid.
Phage-mid.
Yeah.
That's what I'm thinking of. Yeah, phage-mid. So that's a phage plasmid, right?
It's something that is a plasmid that is packaged in the phage head. Yes.
Oh, but it's not like this we're talking about here, which can exist as a plasmid or as a phage.
I don't think so. I'm not sure, but I don't think so.
Okay.
So in the gut, it was originally thought that there were lots of lytic, meaning like actively replicating infections going on, but some recent studies have suggested that the majority of the phage that are found in the gut are temperate and are not actively replicating. So they are present, but they're not making virions that are moving from cell to cell. And the number of these non-replicative phage that are phage plasmids is thought to be anywhere between 5 and 7%, depending on where you look and how you measure it. But they're not easy to culture, so we don't have great information on it. And there are two that we do have information about. One of those is phage P1, which many people are familiar with because it's used to move DNA from one E. coli to another and from E. coli to some other bacteria as well. So phage P1 does exist as a linear double-stranded genome, but it also can circularize and replicate in the cell as an independent episome. And it doesn't integrate into the host genome. That's a really common question I get from the undergrads. This one does not integrate into the host genome, and it is maintained very cleverly as a one copy per cell using multiple control systems. The second one is called, the second phage that is known to exist as a plasmid is called N15. And N15 is a linear plasmid, so I guess this would be more like the parvoviruses. It is linear in its capsids, and then it can circularize in the cell, but it also forms covalently closed ends as a linear plasmid. It also uses two systems for maintaining a low copy number in the cell. The P1 plasmid is about 100, so it's 94 kb, and N15 is about 46 kb. They both have known origins of replication with genes that are associated with that particular type of origin of replication. And these are recognizable by looking at the sequence. So they wanted to apply this bit of knowledge about phage plasmids to this prototypic crAssphage, which according to the ICTV, the International Committee for the Taxonomy of Viruses, has been classified as Cargivirus communis, which I really do prefer the term P-crass because I'm used to it. But so Cargivirus was not able to be cultured as in monoculture because we didn't know the host. has not formed plaques in the past. So in this paper, they then tell us what they find, a few hosts that it can replicate in, and they identify that it has a bunch of phage genes. So the way that they did that is they first took a look at the genome. So they don't have an isolate to start out with, just have sequences all over in the databases. So they took a genome of cargivirus and they looked at the orientation and arrangement of the genes. So if you put it in a circle, you can see that on one side of the circle, most of the genes, or actually all of the genes, are pointed in one direction. And on the other part of the circle, they're all pointed in the other direction. So they're encoded on different strands. The ones that are pointed to the right or on the positive strand are associated with genome replication and things that are associated with plasmids. The ones that are on the left or on the negative strand are structural proteins that are necessary for formation of a virion, as well as other things that are related to lysis of the cell. So that is the side that they call the phage side. And it's probably more like 1/3, 2/3 division. And they notice that at both ends of the plasmid-like genes in the genome, there are replication initiation proteins. And they also found that within that plasmid side of the genome, there are several important proteins, like one that is called MOB-C. So this is a mobilization protein known to be associated with movement of plasmids from one cell into another. So MOB-C is a relaxase that helps to put the plasmid into a conformation that can move from one cell to another. And the replication initiation proteins, there are 2 of them. Usually there's only 1. And so that gave them a clue that there might be some flexibility in the kinds of replication origins that were going to be present and used by the virus.
Jolene,
yeah.
I got to ask you, what's a bacon domain?
So a bacon domain, I don't actually remember what it stands for, but it is a domain in a protein that is thought to be associated with binding to structures on the surface of cells. So it's important for entry.
Got it.
I have to say that this organization somehow is, for me, really satisfying. Okay. I like the fact that this thing looks so organized, that there's a whole section here that's devoted to DNA replication and a whole different section that's devoted to structure. And they're so distinct from each other that they're even transcribed on different strands. It makes you wonder how this came together. It's like a phage, like really a phage-plasmid. Like 2 different things that came together.
But viruses do that,
yeah.
Mitch, right?
Yeah, but this is in the extreme to me. Okay.
They stole something bigger than themselves.
I particularly like the asymmetry in the transcription, that you get all of these genes on one strand in one department and all of the genes on the other strand in the other department. That just makes me happy. I'm not sure why.
Adeno does that too, right?
Adeno does that to a large extent. I think one of the reasons it makes me happy is that phage T7 does that. Phage T7 is all one strand, okay, and very compartmentalized.
So one other gene that they found encodes something that looks to be similar to a protein called GAM, which is found in phage-mu. And this is a protein that binds to the linear ends of the genome of mu when it gets into the cell and protects it from being degraded. So that sort of suggested to them that at some point this genome might be having linear ends available that needed to be protected. So with this, they wanted to look in samples. Remember, they don't have a phage that they can culture and use for studies. So they looked in more publicly available data, and they went to the databases and found both stool metatranscriptomes and metagenomes. So what this allowed them to do is to look at RNA as well as DNA present in those samples. And they looked for samples that had a high coverage of cargivirus, and they found 43 of them. And in those 43, they calculated the expression of the genes across the genome. And they found that that there was a slightly higher average expression or reads per kb million in the plasmid region of the genome compared to the phage region of the genome, which could suggest that you have more of the plasmid genes being expressed, and therefore you're not making so much phage. And they plotted this across all 43 samples, and they show that for basically, I think it's like 38 of them, that they had a higher plasmid gene expression than phage gene expression across those metatranscriptomes.
Yeah, to me, this feels like plasmid is what we do most of the time. But occasionally we go phage on you.
Go phage on you. So that's what they said. This suggested that if it's existing as a plasmid, it's expressing these maintenance proteins that would maintain the certain copies of the plasmid present in the host and make sure it got partitioned into the daughter cells when the host divided. They next looked at origins of replication. So an origin of replication is where the DNA is going to start being copied. And here they thought that they expected, based on what we had seen for P1, that there might be two origins of replication. There would be one that would be for plasmid-like replication and a second one that would be for phage-like replication. This is also something that's pretty common. There's very commonly more than one origin of replication in phage genomes, especially if you have a large phage genome. And here they used a couple of different methods that I'm not super expert in, but they looked at what is called the GC skew across the genome. So they basically took windows of sequence across the genome and they calculated the GC percentage, and then they plotted how much across a certain region was more or less GC. And what that can tell you is when there's a switch in GC, it can tell you that that's where an origin is present and you might start copying. They also compared this to predictions for where an origin is present using some bioinformatic tools that can give you a location where that might be. And they correlated these things together. And what those two things together suggested is that there were 3 positions that might be an origin of replication within the genome. Right. Then they wanted to actually look at some more information related to the conformation of the genome within a sample. So instead of just taking the reads, they wanted to see, what does it look like? Does it look like a circle? Does it look like a line? They had to do something called proxy meta-Hi-C sequencing in stool samples. And before, they were just working with sequences that they had from databases. So here, they had to find a stool sample that had enough CARG virus in it that they could use it. And they did that. They sequenced one and they assembled it, and they found that it was 95.5% similar, or identical, to what they had been using as their reference genome. And that was more similar among the negative strand genes, which are the phage genes, versus the plasmid genes were less conserved. So 97.5% on the negative strand and 90.1% on the positive strand. And the positive strand included those replication proteins that recognize the origin. So they found that sample, and then they did this technique for proxy meta-Hi-C. And so what that means is you have a DNA in a sample or in a cell, and you cross-link the DNA to itself. And so if you have it folded up, then you'll get pieces that are next to each other will be cross-linked to each other, and pieces that are further away from each other will not be cross-linked. And then they cut up the DNA and they circularize the pieces that were next to each other, and then they sequence those. And so that is what allows you to map which positions on a genome are close to other positions on a genome. This is something that's used a lot for mapping the positions in chromosomes of host cells. So here they show that they got very characteristic structure for the genome being more duplicated in certain regions, and for the genome being linear, for the genome being more linear than circular. They saw these repeat sequences. They saw that there were some sequences that were more represented than others, and this suggested that there was a terminal repeat. And that is something that is quite common for linear genomes as well. And so this also gave them an idea that there might be multiple conformations of the genome. Even though they had more representation of what looks like linear genome, they also had some suggestion of the circular conformation.
Yeah, I was really glad that they talked about the repeats at the end, giving them information about a potential linear genome. Again, that kind of reminded me of parvoviruses.
Mm-hmm.
But part of the reason why I really liked that was because I felt very ignorant about this technique and this part of the study. And I was my mind was sort of being blown by them doing this experiment, because I was thinking about, wait, we're measuring the DNA in stool samples and you're expecting it's in its typical conformation and not, degraded. And so my first thought is maybe it's linear because, getting to the point of being in a stool sample, it got linearized. I wasn't thinking about, whether intact cells were coming out or whether the DNA was in its intact form. And so I was sort of uncertain about how to interpret these data, and the fact that the majority was linear. But then when I saw the parts about the potential repeats at the ends,
Mm-hmm.
I was like, oh, I know what that is. I buy it now. So I'm sort of still impressed about the idea that the DNA might be in its sort of actual conformation in the stool sample.
So I have some thoughts about this. First of all, to me, it's very poxy. Okay. That is a linear genome with apparently origins towards the ends. Some repeat sequences, though in POX it's inverted repeats. You mentioned, at least in one of these, covalently closed ends. I don't know that applies in this case. But all of that is pretty poxy, okay, like pox viruses.
Mm-hmm.
But at the same time, the fact that they present these maps sometimes as circles implies to me that in a lot of the sequencing, correct me if I'm wrong, you're finding what we're calling the ends more or less contiguous with each other.
Yeah.
Right?
So,
and all of a sudden I wonder if that doesn't have to be a circle. That could be a concatemer. Right?
Mm-hmm, mm-hmm.
That's also poxy. Okay? It replicates as a linear molecule forming, and it's actually like parvoviruses as well. Replicates as a linear molecule forming concateners that are then resolved into linear molecules.
So the question of whether or not they form covalently closed ends was something that they looked at, but they didn't find a lot of evidence for it. And since that GAM protein is present, which is known to protect the linear ends of DNA, they're thinking it's not so likely to be like N15, which was the one that I mentioned that has the covalently closed ends.
I don't know if you can do this in these samples, but the way you figure that out in pox viruses is to do some sort of either shearing or, better yet, a restriction digest, and then take the whole thing and melt it and quick cool it and isolate the double-stranded DNAs. Because the things that are close, that are covalently closed at the ends, snap back because they're hairpins and form double-stranded DNAs, whereas the pieces from the middle when quick cool don't anneal with each other. My guess is it's such a small fraction of the total that that's not going to work in this case.
Yeah, I think in this case, because it's the stool sample and it's maybe more mixed, it is harder to figure out. But when you can grow the phage in a more homogeneous population, it will be a lot clearer to determine those sorts of details, especially if it's switching conformations at different times during the infection cycle. Okay, so the next thing that they did was they looked at these sequences that were present in public databases, and they looked at genome coverage. And they say that very commonly, if the phage is replicating, then, or if any DNA is replicating, then you will have higher coverage near the initiation of replication than at other places in the genome. And so they did see slightly higher coverage on either side of the terminal repeats, suggesting that there were origins of replications on either side of those repeats. Okay, so then they did look for circles, and they looked for sequences that would go all the way across that direct terminal repeat, that when it's assembled into a circle, would have sequences on both sides. And they said that was always less than 5% of the samples that they had. And so this suggested that although when you assemble a sequence on a computer from sequencing reads, it comes back sometimes as a circle, that doesn't actually mean that it's a circle. And here it looks like less often, well, the great majority of the time, maybe 95% of the time, cargivirus is more in the linear form than in the circular form. And they also looked in those long-read sequencing data for the presence of cargivirus in any host chromosomes. And they looked everywhere in the host genomes, and they did not find any phage sequence in the host chromosome. So this is a negative result, but it supports the idea that it's not integrating into the host chromosome. So speaking of the host, they next wanted to look at what is the host for this phage. And they have a really nice table that summarizes all of the predictions that have been made for cargivirus in terms of what its host is and how that prediction was made. And this spans, all the way back to when it was first reported in 2014 to now. People have done some very creative things, I think, to try to figure out what the host is or are. And one thing they predicted abundance correlation, so how abundant the phage is and how abundant a certain kind of bacterium is. And it's actually not a certain kind of bacterium. These are very high-level classifications of the hosts. And they've looked at homology between the things present in the genome, the phage genome assembly, and the things present in the bacteria. They've looked at oligonucleotide frequency dissimilarity, Bacon homology, CRISPR spacer analysis. So if the host had some sequence that matches to the phage, and that suggests it infected that cell at some point and the cell survived. They've also done co-occurrence in stool fermentations. So because people were trying to figure out what it was infecting, they would take stool, and instead of try to separate out all the individual components, they would just culture in that in a mini fermenter. And so these have given many predicted hosts, anywhere from Prevotelaceae, Bacteroidota, Firmicutes, Proteobacteria, Cyanobacteria. And then there are a few predictions of a specific species, the Phocaeicula, both Vulgatus and Dorii, and a couple of different Bacteroides. We've even got some E. coli in there. And so this tells you that people have really been trying, and you've gotten lots of different answers.
Jillian, I do like the kernelized logistic matrix factorization.
Yeah, I actually don't know what that is.
Well, if a student asks you what it was, what would you tell them?
I would tell them, I am going to look it up. You go read the paper. I'm going to look it up. We'll talk about it next class.
But that's the one that picked up E. coli. It's very
It is the
interesting.
one that picked up E. coli. You're right. I do think that there are some really sophisticated bioinformatic methods that can make these predictions that I probably will never understand the math actually behind them. But anyway, what this lab did was they took an experimental approach. So they found the stool samples that had these cardivirus in them, and they then they grew those. They knew that they couldn't make plaques. They can actually measure it with qPCR, something that's talked a lot about here on TWiV. And what they did was they took this stock where they could culture cardivirus in a stool sample, and they filtered the supernatant so that they could have the phage present. And they then tried growing it on several different individual hosts. Did I skip a section? Yes, I did. So before they did the culturing on individual hosts, they did something very similar to the proxy meta-Hi-C that they had done just to look at the phage genome. And they found that the bacteria and phage DNAs that were close to each other gave them this Phage-Icola vulgatus as one of their hits. They also got Parabacteroides myrdae and Bacteroides stercoris in some samples that they had seen. So then that was why they tried culturing this in P. vulgatus. And they also tested a close relative of P. vulgatus, which was P. dorii. They grew those individually, the way that we would do with most of the isolates that we have in the lab, which is by culturing them to mid-log phase and then applying the filtrate and trying to see an increase in the PFUs while seeing a decrease in the CFUs, or the viable hosts. And what they do not see is that they have a very clear increase in host and a very clear increase in phage and a very clear decrease in host. So if we look at the graph that they have for P. vulgatus, they're measuring this in multiple different ways. They're using the qPCR to get the cardivirus signal, and they see that goes up, kind of. I mean, it does. It goes up. It's not a linear increase, and there is some variability in the increase over time, and this is over more than 40 hours that they're measuring. They also see that the P. vulgatus goes up, but then it goes down. It goes down a lot more than you would expect it to go down without phage. So there is a correlation here. There's like one time point where there's a difference, a significant difference they measure. They say that with phage, there's a lot less CFU, and without phage, there isn't. They do the same thing for P. dorii. They do the same thing for the other hosts that they check that they show in their supplemental. It's not the same as what you see with phage that come in, kill all the hosts, phage numbers go up, host numbers go down. But there is an increase in the number of phage. And that's one of the things that has been noticed before for crassphages. You get an increase in host and you get an increase in phage. And part of that is thought to be because of variations in host surface structures that allows the phage to replicate, but then just be present and not infect, and then the host can replicate and increase, and then the switching of those surface structures allows replication again. So they also look in just media and in supernatant. So before they were looking in whole culture, and here they're separating the media, which is where you would expect released phage to be, and in the pellet, which is where you would expect infected cells to be. So for P. vulgatus, in the pellet, they see that the numbers of cardivirus copies go up. They see that the numbers of phage go up a little bit in the supernatant, and they see that it goes up the same amount in the whole suspension. So this suggests that the increase that they're seeing is because of what's inside of cells, not because of phage that's being released into the media. They see a very similar story with P. dorii, that the majority of the increase seems to be from cargivirus that's intracellular, not cargivirus that is being released into the media. So, all these things together are suggesting that the cargivirus communis can replicate in P. vulgaris and in P. dorii, but it's not releasing phage into the supernatant. It is remaining inside the cell, and it is probably because it is replicating as a plasmid form rather than replicating in a phage form that's going to be packaged into a virion. When they did screening of colonies, they screened over 500 colonies by PCR, and those did not really have a lot of cargivirus in them, which is a little bit surprising. They never were able to make plaques, although they see an increase in cargivirus here. So they may never be able to get plaques. They even they say that they expected that because other people have tried to use something to induce it to make plaques, that they didn't see any of that yield plaques.
Yeah, I would have expected, and I don't recall where they comment on this, that if you were screening colonies, so you take this, they take the stuff that they cultured, right? Mm-hmm. And plate it out and screen colonies. I would expect almost a binary result. Some negative, some like totally positive. Mm-hmm. Because unless ones colonies that start off with a phage-plasmid in them cure themselves as the colony grows. Okay, can you comment on that? That I don't recall if they go into that kind of detail.
I also would have expected them to be able to detect it in the colonies. They did say that. No, actually, I think that was something else. Yeah, I'm really not sure. If the entire culture has increase, I would expect that the majority of the colonies that grow after you plate them should have the presence of the virus sequence. Mm-hmm. I don't know if that's the thing that is difficult for me to be able to, let's see, basically this is all a population level measurement.
Yeah.
Besides, even with the colonies, that's still a population level measurement. Right. What I would really think would help is if we could look at the single cell level, mm-hmm. some sort of flow cytometry or microscopy where you can see what's the prevalence in the population. And partly that's because with the other crassphages that have been, or crAss-like phages that have been cultured, we know that there is significant variation among significant heterogeneity within the population. There may just be only a very low level of infection within the culture, and that is why you only see a small increase when you culture it, even in pure monoculture.
And when they did this culturing, they started off with a clean population of host and a supernatant, a filtrate, correct?
Yes.
So I mean, I'm thinking all the way along that even if the most abundant forms of the genome are a phage-plasmid, that there's probably a background of actually packaged dudes that are floating around, right? Mm-hmm. There may be a minority, but they're there. Does that make sense?
Yes.
Okay.
Yeah, so they see that number goes up a little bit in the media. It's just really low.
Right. Okay.
So do you think that there's enough P. vulgaris in the gut to account for the prevalence of this phage?
Oh, that's a good question.
Or maybe, or do you think there may be other hosts too?
Well, so they do show P. vulgaris and it was B. stercoris, I think, are able to support replication to a low level. So the thing about these kinds of plasmids, especially if it can mobilize between different hosts, as long as it can get into the other host, then it should be fine if there's different kinds of bacteria that can support its replication.
Yeah, I mean, get conjugated into other bacteria, right?
Exactly.
Or whatever. Yeah. So it seems to me that it probably has a broad range. Just that when it infects, maybe it's limited, but the passing of the DNA might not be.
Mm-hmm. Yeah, we don't know if there are host-related factors that could maybe suppress replication in addition to other kinds of things we typically think about for phage defenses.
I mean, you could just take E. coli and put the plasmid DNA. You could transfect it in and see if it would replicate, right?
Yeah, I have no idea if anyone has done that. People have asked if that has been done, and I haven't heard of anyone doing it.
It's pretty simple.
The thing is, the sequences for both transcription and translation in E. coli and a lot of these gut bugs are very different.
Okay. Yeah, that would be
very different. Yeah. So it might work, and that would be a really nice trick. But I wouldn't be surprised if it didn't work. They also do this interesting thing at the end where they're trying to do some calculations to suggest how many the phage get made in an actual lysis event, and they calculate anywhere between 60 to 200 phage per lysis event based on the ratios of cells and phage in their cultures. I'm not entirely convinced. They say that they have single cell analysis, but I think that is something that needs to be measured better. The ones that have been cultured that are not this cargivirus, they do have between 20 to 160 is their burst size. And I think most of the time it's in the right range. So probably correct, but more studies should be done on that. And yeah, overall, the conclusions that they have are that there seems to be, for this very prevalent phage, 2 replication styles. And one of those is that it is present as a plasmid, and that is how it replicates in the host. That is when phage-related genes are being transcribed, and phage particles are unlikely to be getting made during that time. And the other is when the phage side of the genome is activated, particles are made, and then those are spreading via lysis through the population. So I think this is really cool because before, from that table that I was describing, many people have been very interested in determining what is the host. And finding hosts for non-cultured viruses in general is tricky. So the fact that they were able to get some replication in the host by taking into consideration that it might be having this plasmid lifestyle is pretty good.
So I want to double check my understanding of something. So you kind of mentioned the 2 different lifestyles, and both of those can allow DNA to get to another cell in some way.
Mm-hmm.
And whether it's through sort of an extracellular particle or some other, say, conjugation or some kind of transfer.
Mm-hmm. That's right.
Okay.
You know, it's a similar issue with giant viruses. They're often isolated in amoeba, but that doesn't mean that amoeba is the natural host, right?
Mm-hmm.
So
that's true.
people take filtrates of various environmental samples, and they can get the virus in it to grow in an amoeba, but nobody gets the cells from the environment and shows that the virus is in them. It's hard. As you said, it's very difficult to do.
The advantage here is that they used that proxy meta-Hi-C method where they're cross-linking the phage DNA to the host DNA in a stool sample, which isn't the gut, but is how we study most of the things that we have studied about the human gut. Yeah. So that is one, I think, good piece of evidence here in terms of what the host is.
Yeah.
Part of their methods that somehow interested me is a stool culture.
Yes, very interesting.
Okay, so they can take a stool sample and put it in an appropriate medium and culture it anaerobically. It reminds me of years ago when we were on an arc talking about fecal transplants, right, to cure various gut microbe imbalance disorders.
Dysbioses.
Yeah. Dysbioses. Good. And I recall from that, because it really amused me, a machine developed in Guelph for doing large-scale stool culture that they called RoboGut.
Yes.
That was seeded with stool samples. And the culture conditions, must be really tricky. To try and replicate something that is reproducibly over time, like a stool sample.
Yes, I think so.
Thank you, Jolene.
Yeah, this is great. I love the phage stuff. It's great stuff.
This was very impressive paper. They did a lot.
Yeah. And one of the things that impresses me is that most of it is sequencing.
Mm-hmm.
Right?
Or even better, using sequencing someone else paid for.
Yeah. Enormous amount of information out of sequence.
Yeah.
Can you these stool cultures must smell really bad, right?
I
don't know. You don't open them up, man.
Done it.
I guess you got to do it in a hood, then you don't smell it.
I think maybe the collection. I don't know if the collection is different from the sewage samples that we do for phage hunting.
Yeah.
But they're sealed. They're sealed.
Yes.
Yeah. Because they're anaerobic.
You're anaerobic, yeah. But at some point you have to open it. Right?
To do your qPCR. Yeah.
Yeah. So, all right, let's do some picks of the week. Brianne, you're first. What do you have for us?
I have a really interesting article that I read comparing posts about scientific research on Blue Sky versus posts about scientific papers on X. And so this is a Nature news article that is talking about an analysis that was done about how people are posting about science on Blue Sky and X. And one of the reasons why I was interested in this is that I've had a positive experience reading about science on Blue Sky, and I like much of the science content that I'm seeing. And so when I saw that there was a paper actually analyzing this, I was intrigued. And this analysis suggested that research posts on Blue Sky are more original and get better engagement. And so they saw more likes, more reposts, and they also were able to look at the words of the posts and see how close they were to the title. So whether the author was sort of writing something about, this is a cool study that shows blah, 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 or whether they were just restating the title. And it seems as though the posts are sort of more original thoughts about the paper instead of just restating the title. And there seems to be more engagement. So I thought that was pretty cool to see and felt like it went along with my experience that Blue Sky was a pretty good place to talk about science.
Cool. Rich, what do you have?
So there was a flurry of activity, a furry of activity. How about that?
Yeah.
In some of the media outlets about sightings of rabbits in Colorado who had these horny protrusions from their heads, and people were at least interested, if not freaked out by this. Okay, so this made the news, and it caught my attention because there's a very rich history in this. The article describes at least some of this, but doesn't necessarily go into as much detail on the history. So I wanted to, first of all, I think it's a cool disease. Sometimes not cool for the rabbits, but not too bad a lot of times. It is rabbit papillomavirus, or cottontail rabbit papillomavirus, also known as Shope papillomavirus, named after its discoverer, Richard E. Shope, from the 30s. Papillomaviruses are commonly known as viruses that cause skin lesions of one sort or another, very often warts. And these are just huge, extended warts that protrude out of the rabbit's skin, mostly in the head area. And there is some thinking that these creatures have been around forever and are very most common in the this area of Colorado in the West, are the origins of the mythical creature, the jackalope. Okay. Mm-hmm. I am as interested in this because of their discovery, and I put in a wiki article on Shope papillomavirus because Richard Shope was a researcher from the 30s who actually had a lot of input into identifying influenza virus as the source of the 1918 influenza pandemic, but was one of the original virus discoverers and went on to be associated with both this and a rabbit fibroma virus as well. But he was able to demonstrate in the 30s that a cell-free extract of these papillomas would cause papillomas in other rabbits, in recipient rabbits, which stands as one of the first, if not first, demonstrations of a viral origin of a tumor. These are benign tumors, but tumors nevertheless. So that was one of the first suggestions that viruses could cause cancer. Lastly, what I learned about this that was new to me is that there's a whole dynasty of Shopes. Because I just had kind of one Shope in my mind, but this is Richard E. Shope. His son is Robert E. Shope, who is even more famous than Richard E. Shope and was apparently encyclopedic and one of the master virus discoverers down the road. He's got brothers who are also virologists, and I think there's even another generation of Shopes. So when you're talking about Shope, it's not just one person. It's a collection.
Was that information on the Shope family also in Wikipedia?
I had to click around in Wiki. I had to follow links. Okay, yes. I think
Robert has a wiki entry.
Yeah. Robert has a wiki entry. I think I dug into that. You know, when I start poking around in Wiki, it's really a rabbit hole, as it were.
Yeah, I talk about these jackalopes every year in my virology course. The students get a kick out of it. I use that picture that's in the wiki page. Most of those fall off, and the rabbits are okay.
Yeah.
It's cool that it got picked up. Jolene, what do you have for us?
I'm picking a book that I recently read. It's called Lesson Learned: Stories from Women Leaders in STEM. And it's a collection of essays, really, from women scientists across many different fields. There is a significant representation from life sciences, including PhDs and MDs, but there's also a big section on women in technology and women in engineering, which I found to be pretty enlightening in terms of their stories, their journeys, the lessons they feel that they have learned as leaders. And one of my favorite parts that I read was a little essay from Rachel Willand Charnley, who is a PhD in chemistry. And she has this analogy that I loved. She says, being an underrepresented minority in STEM is akin to being on an energy diagram. a reactant in a long series of synthetic transformations, constantly transitioning to become a new product. We enter the reaction and immediately begin to climb the activation energy barrier to transition to a lower energy product, an undergraduate, a graduate, a postdoc, tenure track professor, a tenured professor. And she says, some of the reactants I interacted with built me up, forming strong bonds, contributing to the formation of necessary functional groups, while others broke bonds and contributed to a re-optimized synthetic roadmap. And I love that analogy. There are lots of interesting things that they have to say in here. It's not really advice. It's just kind of a remember that everybody has a different story. Everybody has some struggles. Nobody has a perfect balance in their life, but they're all scientists and they've all accomplished a lot. And so the idea was to have stories that other women could look up to realize that they could have a journey into science as well.
Very cool.
Nice.
I like that.
I like that very much. So many years ago, 4 or 5 years ago, I picked Adam Neely's YouTube channel. Adam Neely is a musician who explains music. And I remember my pick was he was explaining The Girl from Ipanema, this iconic song, and what was unusual about it, right? And so now I have another similar pick, although this one is just rock and roll. This is Rick Beato's channel on YouTube. And he does a lot of, he's a guitarist. He's really a good guitarist. And he does a lot of interviews and things of that sort. But he does a series called What Makes This Song Great? And there are all kinds of songs there. But the one I stumbled upon was Smells Like Teen Spirit by Nirvana. Because I wanted to play it. So I just looked it up and his thing popped up. And he just takes it apart in a beautiful way and explains to you why it just sounds great, right? It just sounds really good. It's got an atmospheric sense to it. And because Kurt Cobain, who plays the guitar, just did everything perfectly. He said he was a brilliant melodicist, but he didn't know what he was doing, actually. So he goes through all these chord theories and explains them. And he says, Kurt didn't know any of this. He just felt it. He just knew how to make music that really sounded good. And he talks about the lead guitar, the bass line, and the drummer, Dave Grohl, right, who's since become very famous, and how they all come together to make this incredible song. He does it for many other songs as well, and it's really good. But I love that he's just sitting in his studio, and he's talking pretty much off the cuff. He's got his guitar there, and he plays parts of the songs, and he explains it. He plays the actual song itself. It's a lot of fun. I
will have to have a look at this. Frankly, I looked at your pick, but I wound up clicking on the Adam Neely channel to start with.
Well, Adam is great. Yeah,
and
really good.
Wow, what a music nerd he is. Unbelievable.
He's mainly jazz and classical, although a little bit of rock in there. But both of these guys understand their music theory.
Yeah, really it's good. Great. Well, it reminds me, I sing with a barbershop harmony group that now has like 7 people in it or something like that. We've been singing together for like 4 years now, and we rehearse on a regular basis. And the rehearsals have become pretty sophisticated. So I mean, a lot of us, myself included, aren't real musicians, but there are a couple who are. So we'll sing a song, and then the discussion afterwards that goes on for 5 minutes or so is, this sounds really cool. Why?
Yes.
Okay. Yeah. And there'll be a whole bunch of analysis of the chord and that kind of stuff. It's just great fun, great stuff. I wish I understood 1/10 of it.
I'm super excited to watch this.
It's good.
And it's going to make me feel good that I don't really like the song just because of sort of my age bracket. That I'm in just the right age for this song, but it's a good song too.
He also talks about the history behind this. So this song changed rock and roll.
I will have a look at this. This
completely changed it. The number one hit at the time was something by Michael Jackson, and this blew it away. It's just amazing. But he does other good stuff too. He recently he discussed the top 10 Spotify. So now it doesn't matter how many albums. Nobody sells albums anymore. It used to be if you sold, 1 million albums, you'd get something. but now it's streams, right? You have to have billions of streams. So the top 10 Spotify, he went through them all and gave his comments on each. That's really good. I like that as well. So we have 2 listener picks. Lori writes, my pick is, I recently saw this video on YouTube. Thought you and your listeners might enjoy it. Who knew bats could be so cute? So this is the Tolga Bat Hospital. And it's a bat hospital, and it's a video of what they do with bats, feed them and take care of them. And they're very cute.
Yeah, I watched this. It's very nice.
And Amit has an author recommendation. A PhD in zoology and resource ecology turned writer. He decided to write hard science fiction. The Rifter, this is probably for you, Rich. The Rifter trilogy is the most relevant to virology, but it's also the largest assault on the reader's sensibilities. Subject matters not for the faint of heart. Blindsight and Echopraxia are his stronger standout works, allegedly getting adaptations in the near future. He includes pages of citations for most of the science referenced in his books, creating a research rabbit hole. Excellent science fiction and unnervingly predictive, but difficult to recommend to anyone without a science background. So
I think the author's name is Peter Watts. I'm not sure if he said that.
Peter Watts. Yeah.
Emmett, why couldn't you write this and send it to me at the beginning of the semester instead of at the end of the first week of class? I really want to read these, and I don't know when that's going to happen.
You'll find some time, Brianna.
I used to read most of my science fiction when I was supposed to be studying for exams in graduate school.
I know a guy who's a physics professor at Cornell, and he said when he went to Princeton, and he said in Princeton they have a study break over the holidays so you can study for your exams. He said, I spent the whole break reading the trilogy by Tolkien. Sounds good. And that is TWiV 1249. Show notes are at microbe.tv/TWiV if you want to send us questions or comments or picks of the week. We love to hear your picks. Send them in. TWiV at microbe.tv. And if you like these programs, we'd love your support. We have 8 other science podcasts at Microbe TV. Go over to microbe.tv/contribute. Jolene Ramsey, Texas A&M University. Thank you, Jolene.
Thanks. I enjoyed talking about phage and herpes virus.
CrAssphage. What a name, huh? I think they did that on purpose, right?
Oh, yeah.
There are a lot of, stories about where the name come from and what it actually means.
Brianne Barker's at Drew University. Bio Prof. Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Rich Condit is an emeritus professor, University of Florida. Gainesville, he's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. I love being with you folks and doing this. This is great.
Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.